This is Stanford Engineering's The Future of Everything, and I'm your host, Russ Altman. If you're enjoying the show, or if it's helped you in any way, please consider rating and reviewing it to share your thoughts. Your input is extremely valuable, and it'll help others learn about the show. Today, Professor Sergiu Pasca from Stanford University will tell us how his group has figured out how to take skin cells, turn them into brain cells, and then study some of the most vexing psychiatric and neurological diseases in a dish with the idea of generating new therapies. It's the future of brain science. So, neurological and psychiatric diseases are some of the hardest diseases to study in the lab and to treat. And those are related. Because we don't have good ways to study the brain, nobody wants to give up a piece of their brain for scientific research. It's really hard to understand what goes wrong at the molecular and cellular level and how we can invent new ways to treat terrible diseases such as chronic pain, schizophrenia, bipolar disorder, and depression. Well, Professor Sergio Pasca is a professor of psychiatry and behavioral sciences at Stanford University, and his group has figured out how to take skin cells, make them go backwards in time to become stem cells, which have many potential future fates, and then get those stem cells to differentiate into little pieces of brain. His group has shown that they can do this reliably and they can generate little circuits where the cells talk to one another and recapitulate some of the deficits that are seen in patients who have certain diseases. It's an amazing story, and you're about to hear about it. Sergio, you've developed this amazing ability to grow living tissue that mimics parts of the human brain. First of all, how does that work? Then we can talk about why you did it and what we can use it for. But tell me, how do we grow little pieces of brain?
Well, the process starts by generally taking a tiny skin biopsy. So just taking a few cells from the skin of any individual, say a patient with autism or with schizophrenia, bringing those cells into the lab, and then essentially doing some mumbo jumbo to the cells, playing cellular alchemy and pushing those cells back in time to essentially turn into stem cell like cells.
Uh-huh.
And the reason why we do this is because stem cells have this remarkable ability of turning into any other cell types. So once you have the stem cells from any individual, not only can you store them and share them with others, but now you can gently guide them in a dish to become brain cells. And we spent essentially the past 15 years designing strategies to turn these stem cells from any individual into various types of brain stems and brain cells into various ever more complicated preparations.
Fantastic. So, a couple of questions to follow up. I think everybody knows that part of what makes the brain is there's a very complicated architecture of the cells, where they are, how they interact with one another. Are you able to get that to happen, or are these just individual cells sitting in a dish but not talking to each other and not creating the kinds of structures that we see when we do, studies of brain tissue?
That's a very good question. So about 15 years or so ago, we were doing exactly that, kind of like at the bottom of a dish, a flat layer of brain cells, let's say cortical neurons, so neurons of the cerebral cortex. Of course, they were great. I mean, they look like neurons. They're human neurons. Their shape and their properties were those of human neurons, but they're not able to communicate with each other the way they do in the three-dimensional brain. And more importantly, as you pointed out, they're not able to communicate with neurons at a distance. Because what makes the human brain unique, or the brain really unique, is that unlike other organs that are more homogeneous, let's say the liver, it does matter which part you're actually probing or you're looking at. And so what we've actually slowly done is, first of all, generate three-dimensional preparations that are now known as organoids that resemble parts of the nervous system, let's say parts of the spinal cord or parts of the cerebral cortex. And that we spent, 5, 6, 7 years, kept refining those methods. And at one point, we really became interested in studying cell-cell interactions. We thought that was important, that forming circuits from the cells will be enabling for so many applications. Yeah. So then we introduced what are now known as assembloids. Assembloids, which are essentially assembled three-dimensional cultures, where, let's say, you make a three-dimensional culture that resembles the cerebral cortex and one that resembles the spinal cord. And then you culture them separately. You provide different cues and small molecules so that they become those cells. And then at the right time, which, depends a little bit on the brain region, you start putting them together. And by putting them together, literally you put them in physical proximity to each other.
Yes.
And initially we thought we're going to have to guide and we're going to have to tell the cells what to do, kind of like in engineering, where you want to provide the blueprint. Of course, what we got wrong at that time was in biology, the cells already know what to do. So once you actually put them together, there are these emergent properties. The cells will recognize where they actually have to go and they'll start project and find, for instance, motor neurons, connect with them. And then if you put a third part, let's say a piece of muscle that you get from a biopsy, then those neurons will connect to the muscle. And suddenly the muscle just starts twitching in a dish under the control of cortical neurons. So I was really exciting because suddenly you can witness the remarkable power of self-organization for a process that, to be honest, we don't even understand that well.
Right.
It's not like we know how the rules of assembly of those, but once you provide sort of the minimal conditions necessary, you unleash these forces of self-organization.
So the cells really want to be a brain. They want to be a spinal cord, and you just have to let them do it almost.
Precisely.
So one final preparatory question is, when you're creating these individual organoids or that will become part of an assembloid, does it always have to be just one type of cell? You talked about livers, and even livers, as we know, are made out of multiple cell types. So how advanced is your ability to make a mix of cells, for example, the cells that are feeding the neurons or that are supporting the infrastructure of the brain, or is that a future topic?
Well, I mean, one way to look at brain developments is that there are multiple niches, there are multiple places where cells are born. And those niches will have cues for those stem cells to generate the diversity of cells. They're all kind of coming from there. So let's take the example of the cerebral cortex, the outer layer of the brain, the region of the brain where, we think that all of our cognition and what makes us human comes from. Most of the cells in the cerebral cortex are born in a region close to the ventricle, a ventricular zone, where there are these progenitors called radioglia. This radioglia will essentially generate almost all the cells in the cerebral cortex in a sequential manner. Uh-huh. For instance, they'll first make deep layers of the cortex and then upper layer of the cortex, and the cells will organize themselves in a sequential manner. So as you can imagine, if you specify the generation of this particular type of stem cells, you can expect to generate all the cells in the region.
Fantastic.
Now, that doesn't mean that you get all of them, because some of the cells do come from outside of the nervous system. So, for instance, microglia, the resident immune cells of the brain, are not even born in the brain. They're born far away. They're mesodermal in origin. So if you really want to put them in, you have to make them separately and then insert them into this preparation.
Okay.
And so that becomes, again, an assembloid to a large extent.
Okay, great. Well, I could go on forever, but that has set us up beautifully for the conversation, which is, why did you do this, and what are you using these brain tissues to do?
Well, deep in core, I think what I am is a frustrated physician, and that's what I think I've been, and I continue to be. And for me, as I was doing my medical training, I was, in awe of what molecular biology can do for most branches of medicine, as you very well know. And, I was training at a time where we started to see some of the first rationally designed drugs in oncology, where literally, you weren't just hitting all the cells, so that they would all die, but you will have a pathway and you'll have a targeted therapeutic that will just cure the cancer. And then, you go on the, oncology, and then you move and you go in psychiatry to see patients. And it's almost like a completely different world. Their disorders are not defined molecularly or cellularly in any way. They're defined behaviorally. And they're, also most of the drugs that we already have not been designed rationally, have not been discovered because we understand the pathophysiology, but rather in reverse. They're found by chance, and then we've kind of tried to figure out how they work and what the disease is like based on that. And that was really frustrating for me, I must say. And it became clear that the reason why psychiatry and neurology have been left behind versus all other branches of medicine is to a large extent because we cannot access the human brain.
Right. People don't usually want to give you a sample of their brain to study.
They don't.
For
And
understandable reasons.
for very understandable reasons, and nor do they give you one to test a drug, before you do it. And that was really frustrating for me at that time. And I was doing a lot of experiments in animals, cat and rats and animals. And it was incredibly powerful to be able to like stick an electrode into the brain of an animal and listen to electrical activity, right? As the animal was performing various tasks. And it becomes clear that if we really want to understand these neuropsychiatric diseases, ultimately we will need to listen very carefully, very closely to those neurons and understand the molecular basis for that. And at that time, as I was finishing my clinical training, there was this remarkable breakthrough that was published around that time that certainly still sounded as science fiction. A lot of people didn't believe that it was true, which was this idea that you could take any cell, a skin cell, and turn it into a stem cell. And it was, as you can imagine, outrageous because everybody thought, and it was dogma at that time, that development is a one-way street. You can never go back. Once a cell has been formed, you just don't go back to become a stem cell. It would be a liability to like forming cancers. And yet, Yamanaka showed convincingly that you could do that. And in my mind, that kind of like clicked and realized that it would be a remarkable opportunity for us in psychiatry to form, to build brain cells from individuals in a non-invasive way without really accessing their brains. And then essentially derailed my career to a large extent. And I sort of like gave myself 15 years of like trying to do basic science, hoping that, we'll make some advances. And I'm coming very close to those 15 years. So we'll see how it goes. But for me, it really came out of this frustration that I felt we were making much biological progress in understanding this.
Okay, so let's go right to it. So if you take the skin of somebody who has, let's say, bipolar disorder or schizophrenia, and if you make a little piece of brain, it's not the whole brain, it's a little piece, can you tell that there are problems with those cells that might relate to the fact that the patient has schizophrenia? Because that would be remarkable, because then you could sprinkle drugs on top of it. I use that as a metaphor, of course, and see, does that fix whatever is wrong? So does a schizophrenic patient, when they go through this process of giving you their skin cells, do you wind up with little pieces of brain that have relevance to schizophrenia?
Yeah. Well, the challenge with that approach generally is that we wouldn't even really know what to look for.
Right.
Like for a patient with bipolar, how would we know that we found something? And that was really the challenge in the beginning, because a lot of people said like, well, sure, you can make the cells, but how are we going to know? So actually choosing the disease that you study judiciously is actually crucial. So what we actually did is we anchored it in genetics, actually. We went back and we thought, well, there are genetic forms of psychiatric disease, highly penetrant disorders where if you have this mutation, you'll certainly be very sick. And some of those mutations are in genes that we kind of like know what they do overall. So we could test the function of those cells. And the classic example, and the one that we focused on 15 years ago, is this rare form of autism and epilepsy called Timothy syndrome, which is caused by a mutation in a calcium channel. And calcium channel, as the name implies, they carry calcium inside the cells. So we thought, if we're going to be able to make neurons from these patients, we could actually measure how calcium goes inside the cells. And if there's something abnormal, we'll know what to look for.
Right. So you have the hook there that you know what you're expecting, where the abnormality might be.
Precisely. At least we have some sort of, benchmarking that we can do. Not that we would know what would come afterwards, but it would tell us at least that there is something valid about these models. And that's exactly what we did early on. And then, of course, continued characterizing the cells. And you can find all kinds of like defects now we know. Not just in how the cells handle calcium, but how the cells are moving, for instance, throughout the nervous system, how they're making connections with each other. And once you find some of these defects, whether they're like in organoids or in assembloids, then you can really start contemplating the idea, let's try to reverse them. Let's try to sprinkle specific drugs or, find other therapeutics to reverse them, with the idea that if you were to reverse those in a dish or in some of these models, they will have an effect in patients. And of course, that step has not yet been done. Nobody has really went all the way. We're getting close, but nobody has really gone all the way to saying, okay, we'll reverse this now in a dish. let's move into a patient and show that this will work.
Fantastic. Okay. So you have some big news coming up around now. Can you give us a little flavor as we close this first segment? What's the latest things that coming out of the lab that you're most excited about?
Yeah, I mean, for the past 10, 15 years, essentially what we did, I feel, is been building tools to a large extent. And paradoxically, my lab is probably much more well known as a tool building lab than I wish. I don't consider myself a tool builder. I never really wanted to build tools, to be honest. I wanted to understand the pathophysiology of disease. But all these years of actually using these novel tools, or trying to understand the biology of these disorders, are starting to pan out, in the sense that for Timothy syndrome in particular, because this is the paper that is coming out next week, we got such a good understanding of the biology of this condition through these human cellular models, organoids, assembloids, 2D cultures, that at one point the therapeutic opportunity just became self-evident.
Wow.
It just became clear this is exactly what we need to do. And this came like 3-4 years ago. And then through a series of experiments that we've done, we've now developed a strategy that can essentially reverse almost all the phenotypes, all the defects that we've discovered in patient cells over the years. Literally some of them within hours, some of them within days, but they're essentially just like completely reversing them. And I think that's very powerful. And of course, it poses all kinds of questions of like, what do you do next? There's not a very good animal model for this disease. So how do you test before you go into patients? And that's where another tool that we were developing in parallel came to help, which is, most of these therapeutics really do need to be tested in a living organism, in vivo.
Yes, of course.
Rather than just in a dish. It's very important. We know that already. A lot of failures come from the fact that something works really well in a dish. You go in a living organism, it doesn't work, right, or it has like unanticipated side effects. Yes. So how do you do that? How do you test something, while keeping the patient safe? How do you test it on human patient cells without harming the patient? So a few years ago, we devised a strategy where we actually can take some of these organoids, but then transplant them into animals. We put them into a rat early in development so that you essentially build a unit of human cortex into that rat. So almost a third of a rat hemisphere will essentially have human tissue.
Hemisphere of its brain.
Of a rat, yeah. Yes. So you can do an MRI and you can see that a third of a hemisphere now has human tissue that we transplanted there early on. And it's vascularized. It receives sensory input. You can move the whiskers of the
I'm going to bookmark putting a human tissue, brain tissue into a rat for later on in our discussion, but I just wanted to bookmark that. Keep going.
But essentially now the human cells, the human neurons, are integrated into the rat circuitry.
Yes.
And the reason why that is important is because now if you have a therapeutic for that disease, you can come and inject it into the rat, so in an in vivo system, but then look at the effect on human cells.
Yep.
And you can envision that it could be exactly the patient that you plan on treating.
Yep.
Right. And then a week later, you can get out the tissue and say, has there been an effect? Is the drug working? Is it having unanticipated side effects like an immune reaction or other? So I think that is a very powerful way to actually test these drugs before we move into the clinic.
Yes.
And so that's one of the things that we've done for Timothy syndrome in particular, as we've had all these in vitro studies. At one point, we were like, what is next? What are we going to do? How are we going to get into...
And for just to finish up for this part of the conversation, do the rats that you generated have brain tissue from patients' skin who had Timothy syndrome?
Exactly.
So you've made the... Well, this is The Future of Everything with Russ Altman. More with Sergiu Pasca next. Welcome back to The Future of Everything. This is Russ Altman, and I'm speaking with Professor Sergiu Pasca from Stanford University. In the last segment, Sergio told us about his amazing ability to take skin cells and turn them into small pieces of human brain tissue that can be studied for disease and for new therapies. In this segment, he'll tell us how complex these brain circuits can get and some new results showing that they're even able to model complex circuits like the perception of pain. He's also going to address the ethical issues about manipulating what are peculiar to humans, brain cells. And also he'll talk about how his group is sharing their technologies so that brain scientists all over the world can benefit. Sergio, there's a lot of things that go wrong in the brain, and it's not always as simple. It's sometimes a complex set of connections between cells. They call them like neural circuits. So where is our ability to engineer these more complex communication pathways that are important to understand certain diseases?
You're right on point, because most psychiatric disorders are likely going to arise not from, drastic changes to cell composition of the brain. It's not like parts of the nervous system are missing, but rather arise from faulty communication between neurons in specific circuits. So it's becoming really important that with our human cell models, we're building ever more complex circuitry in a dish that allows us to probe some of this mechanism. So, initially the assembloids that we were building had just like two parts. You would just put them in close proximity. Cells will migrate from one part to the other, or they'll project.
Right.
Then, of course, we wanted to put like more parts because most of the circuits have more components. So we put three parts: put a cortex, a spinal cord, the muscle. But then, of course, some of these pathways have more than three. And actually, adding a fourth component was not trivial. It took several years to create both the components and, kind of like the technology that is necessary to grow some of these cultures for longer periods of time. And just to give you an example of something that we've put together quite recently, has been to actually build a sensory pathway, a somatosensory pathway from four parts.
Oh.
And so somatosensory or somatosensation essentially sends information from the periphery to the nervous system. So for instance, tactile information or pain information or temperature is sensed by terminals in our skin that send those signals to the spinal cord. The spinal cord is projected all the way in the middle of the brain in a structure called the thalamus. And then the thalamus will send it to the cortex, where that sensation ultimately arises. And so there are multiple components. So we spent several years making each of these components of the circuit, and then we put them together. And recently we've succeeded in assembling the entire pathway.
Wow.
That actually can respond to pain-like stimuli. So you can actually make a sensory organoid, a spinal cord organoid, a thalamus organoid, and a cortical organoid. You put them together and form a somatosensory assembloid.
So earlier in our conversation, you were telling us about this amazing capability for the cells. You just have to give them the chance to be successful, and they figure out what to do. In this case of this 4 different cells, was that still the case, that they knew what to do, or do things get sufficiently complicated that you kind of have to nudge them a little bit more than you do in the previous cases?
No, actually, again, they know what to do.
Wow.
I mean, they know what to do, and we wouldn't know how to instruct them, to be honest. We're still like pretty ignorant about this. But some of the circuits are so ancestral and so well conserved that probably their evolutionary mechanism in place to establish some of that basic connectivity almost intrinsically.
And I find this so exciting because based on your story with the Timothy syndrome, you now can create pain or create touch, and then you can try to manipulate with drugs to see if you can modify the experience in the dish, and then presumably modify the experience in the rat and then the humans. Is that the kind of plan you imagine?
Yeah, absolutely. And it's quite interesting because once you put all these 4 components together, they connect with each other. And the reason why we know they're connecting with each other is because if you look at the activity of the neurons across the 4 components, they become synchronized. Rather than each kind of like sparkling their own kind of like song, suddenly they're all talking to each other, and you see this emergent property, this emergent activity of the 4-part component. And then what you can do is you can sprinkle in some of the stimuli that we know are mimicking, let's say, pain. So, one
like
thing
hot
that
pepper.
people like, exactly. So you can take capsaicin from like hot pepper. And if you sprinkle that, which by the way, if you add it even to your skin, it's painful.
Yes,
but
it hurts.
If you add it here, then you'll suddenly just see that the first component lights up, the sensory one, and it transmits activity throughout the entire pathway. So that gives us, some indication that indeed it actually works. And then what you can do even more is you can now model diseases. And so there are mutations that cause either loss of pain
Yes.
or exacerbation of pain,
Yes.
both of which are bad, obviously. And you can induce those mutations. They're generally in sodium channels. You can actually induce those mutations in this preparation and suddenly see that there's either, desynchronization of activities, so the sensory systems are not, processing the stimulus right, or there's over processing. So it gives us really for the first time the ability to watch this communication between the sensory neurons and the circuitry
Yes.
live in front of our eye, but with human cells.
And I can see how the adding of 4, it's just the beginning, because anybody who's a clinician knows that after patients are exposed to pain chronically, they start to get psychiatric responses to the pain. Part of the pain is their high level processing of the experience of pain. I've even had guests on the show talking about these, and you can imagine that you're going to need to build level 5, 6, and 7 so that we can start to understand how those raw stimulation of pain leads to changes in how you process information at a much higher level. I mean, I don't mean to give you any homework, but I could imagine that this could be on the list.
Absolutely. And right now, our system, and I think this is important to clarify also so to not create any confusion, is the vallus of pain, the sensation of pain. It's usually processed in other brain regions. Right. This information is taken from the cortex and is processed, and it gives us the sensation of pain. We don't have that yet in the system.
Right.
That will have to be built, exactly as you said, separately. And in fact, many of the circuits are loop circuits. They're going up in loop, which is really important. And so we've been working quite diligently in trying to build ever more complex circuits. Of course, they're all in vitro, so they're never going to have the full, complexity of an in vivo circuit. But we also don't want the full complexity. Yes. We want the system to be sufficiently complex yet simple for us to dissect the molecular components and move forward therapeutically.
So as I promised a few minutes ago, I definitely want to go to the ethics of this, because the headline here, well, the headline is really that you're addressing diseases that we didn't have an ability to address before. But another headline is that we're taking pieces of human brain cells and we're putting them in animals, and basically you're manipulating a lot of things that some people may consider very sacrosanct, very human, and very peculiar to humans. So tell me a little bit about how, as a scientist and a physician, you have navigated some of the ethical issues about making sure that your work is accepted and that people understand what you are doing, and if you're drawing any lines, where you're drawing those. What's the situation there?
Yeah. No, this is a very important component. To be honest, I never really kind of thought or planned for it as we were moving forward. But, the paradox is as follows. Psychiatric disorders are to a large extent uniquely human. Or, most of them, if not all of them, are uniquely human. And so, as you can imagine, if you want to understand this, the models that we have for those disorders need to be as human as possible. And yet, the more human they become, the more uncomfortable we feel about creating something that is really human-like experience. Yeah. And so, certainly, we're not there with any of the preparations that we have, but as our preparations are getting ever more complex, there are all kinds of concerns that are being raised about this. Some of them, very careful ones, some of them anchored in misunderstanding of what it's actually done. But I think that is exactly the moment when you have to have conversations with, first of all, the broader scientific community, and then with the public itself. I think the public has a responsibility to be part of these conversations. Most of this work is paid by federal grants. And I think the general public has the right to be involved in this conversation.
What kind of feedback have you gotten? Oh, I'm sure you've given popular talks to the lay public. How do they respond?
So generally, I think the public understands really well what the motivation. I think if the motivation comes through very clearly, I think it's everybody will connect. Everybody knows somebody who's suffering from a psychiatric disease, right? I mean, these are affecting 1 in 5 individuals. And everybody knows just how devastating these conditions are and that we will need some solutions. So I think people will understand. But at the same time, this type of research needs to be done responsibly because we are crossing various boundaries. You could say by building ever more complex circuitry. And I think in vitro, there are fewer concerns that I think are realistic in the sense that, sure, the circuit can get very complex, but there's no sensory input that is there's no output that is meaningful, or it's very difficult to do both. But you could envision that if you transplant into an animal, there is another series of controversies that are arising. So what I've done is, first of all, we've had a lot of conversations with others. I gave a series of public talks, including a TED Talk, which was a really important component of engaging already the public. And just recently, I co-organized a meeting at Asilomar in kind of like the historical site where some of this conversation actually started, with like Berg doing
The
solo.
1970s, when genetic engineering became possible. Yes.
Yeah. So together with my colleague here at Stanford, Hank Greeley, we co-organized this meeting at Asilomar, where we brought together not just scientists from the field, but actually scientists from outside the field, people who are studying consciousness, philosophers, evolutionary biologists, social scientists, to really think, very openly about this. And we're putting together a series of guidelines. We're in the process of like writing up that hopefully will catalyze further discussions. And our hope is that in the future we'll have an even bigger meeting that will engage even more broadly, families of patients, but other scientists across the field, and neuroscientists in particular, as we think about these issues.
Fantastic. Well, we're almost out of time, but I wanted to give you 30 seconds to talk about the very exciting prospect that you are now sharing this technology so that others can use it. And just give me a 15, 20 seconds on what's the idea about sharing these technologies.
Yeah, well, I realized very quickly that as we were developing technologies, and again, as I mentioned, I never envisioned being a tool developer. I don't consider myself a tool developer. But at one point, once we've had, half a dozen or a dozen of these techniques put out, there are a lot of people who wanted to implement them. There are a lot of like troubleshooting, and these experiments are not trivial. Unlike like CRISPR, where, you can do your experiments like very quickly in 2-3 days and maybe even try them in the kitchen at home. These experiments require hundreds of days.
Not for the kitchen.
And there's a lot of troubleshooting, and not in the kitchen. And so they require a lot of effort. And initially we've helped individual labs one by one. I mean, literally more than 100 labs, helping them to implement step by step the technique. At one point it became overwhelming. And so what we did is, through the center that I lead here at Stanford, the Stanford Brain Organogenesis Center, we started putting together a course, an international course, where we would bring, 25 students at a time and teach them essentially all the tricks of the trade within 1 week. And we prepare, of course, for months before, because it's kind of like a cooking show, I mean, we have all the components ready. They do the critical steps. They would never be able to do 100-day long experiments in a week, but they learn the critical steps. And when they come here, they also make a pledge that they will go back home and they will teach others at their own institutions. And that has, beautiful amplifying effects, because now there are hundreds and hundreds of alumni. I mean, I travel around the world and people come and say, oh, we've learned at the course, or we've learned from somebody who learned at the course. So it's been really remarkable. And there's so much to do in neurology and psychiatry for a lifetime. So, and I think here at Stanford, we've always had a culture of like broadly and openly sharing whatever we developed early. So it really just aligned beautifully, I think, with the philosophy that we have here.
Well, what a great way to end. Thanks to Sergiu Pasca. That was the future of brain science. Thanks for tuning into this episode. With over 250 episodes back in our archive, you can have instant access to lots of good conversations with brilliant people passionate about their work. If you're enjoying the show, or if it helps you in any way, the bar is pretty low there, please rate and review and give it a 5. You can connect with me on X or Twitter at RB Altman, and you can follow Stanford Engineering at Stanford ENG.